Okay, cool. Let me just. Joseph, you can record because you're the host.
Yeah, I started recording.
Okay, great. All right, so yeah, hi everyone. So we're just going to go over some slides on what Igem is, what synthetic biology is, and what we're planning to do. So just a brief agenda. Quick introduction, a bit about what we did last year and what we're going to do this year. And then I guess we can open up for some questions. Then we're going to have an election for the internal communications right after this. Right. So what is synthetic biology? Um, so I guess if you heard of iGEM, I guess you should have some sort of notion of what synthetic biology is. But in general, it's sort of this way of thinking about cells as factories and as molecular machines, and thinking about them as input-output devices. So this is really a nice abstraction to think about stuff like genetic circuits that can make logic gates, proteins that essentially can convert one metabolite to another. So there's many different applications um, of this, and in particular, what we're working on is dealing with just proteins and designing them. So there's this competition called iGEM that started in the early 2000s, and it basically is like this international competition where teams from all over the world come together, usually in Boston this year. They say it's going to be in Paris, but who knows given the pandemic. Um, so each team comes up with a biological solution to some real world problem. For example, dealing with plastic pollution was our problem for 2019. And for example, some French team came up with a way to detect disease in vineyards. So importantly, it's a combination of wet lab, dry lab, and policy and practices. So not only do you have to have some implemented synthetic biology product, you should also model it with mathematical modeling that can guide this design. So talk with stakeholders, collaborate with other teams, etc. And ultimately, we present our work at the Giant Jamboree in October, November. And in this year, actually, we're going to consider also submitting our work as a preprint. So just to give you an idea of the legitimacy of iGEM, recently there's been this, there was this preprint, and it shows the number of publications for projects that came out or started as an iGEM project. So really this growth is quite significant, and it really goes to show how great of a research environment participating in iGEM is, in particular because really you can be creative, you can fail in a controlled environment, and it gives you everything that you need to run basically a research project in an interdisciplinary environment. So here's just some examples of past iGEM projects, and you can take a look at these on your own time, but their websites are really stunning, and they're just great ideas, all of these and many more. So here. Oh, hello, can you hear me?
Yeah, you cut out for a bit.
Okay. Yeah, so basically the problem, as I said, the problem we were dealing with is plastic pollution. And really it's ubiquitous, unfortunately. Plastic waste in terms of these beads, these microplastics ending up in all sorts of environments. So really our goal is to see, well, what is the potential biological solution that could deal with this problem? This problem really is also exacerbated by the recent, more contemporary situation with COVID-19 and the acceleration of production of PPE, masks, and takeout containers. All of these are contributing to the accumulation of plastic waste. So our project for the past 2 years has been to design or optimize an enzyme with the ability to break down a particular type of plastic. So what you see in front of you is a stereo view of the protein we were working with called PETase, PETase, standing for an enzyme that can degrade the PET plastic. So the idea is we had this enzyme, some team in Japan discovered this enzyme in bacteria, and the problem is that it's really slow at degrading plastic. So what we can do, or our goal was to try to optimize the rate of plastic degradation using a combination of rational design approaches and the machine learning approaches. So then after designing these proteins and then we're gonna synthesize them in the lab. In 2019, we did have the opportunity to do that. And we found that some of our rational designs and even some of our machine learning designs were really promising. And they showed better activity in the wild type, and they were competitive with the current best design, at least of that time. Recently, there have been a lot more designs and a lot more work on this. So we're really excited to continue this project into our third year. Yeah, Joseph, did you want to talk about the plan for this year?
Yeah, for sure. So as Tianyu eloquently put it, we're continuing the project from the last 2 years. But this year, we are really trying to take kind of all of the learning experiences from the first year and all of the hard work in computational research that we did last year and really apply it into the lab to validate our design. So we begin obviously from January and February recruiting our team. Then in March, we essentially onboard all of our team members, get them on the same page, do a bit of a literature review so everyone knows where we're going. And we start our first round of machine learning and genetic construct design. Then in April, we take a break for exams, obviously. And in May, we begin our first work in the lab. What is really cool this year is that we're partnering up with the Canadian Synthetic Biology Educational Research Group called CSBERG, and they're going to give us onboarding lectures on synthetic biology, on business, and on computational biology, so that our members kind of have a really good base from which they can work on. And in the lab in May, the first goal will be to kind of clone and then purify the sequences from last year. Then in June, the dry lab team should have their first batch of new sequences that they've designed. And the goal for the wet lab team will be to integrate these variants into a microbial chasis, as well as doing an in vitro assay to determine their activity and thermostability. After we determine those, the information based on the info on those sequences, we'll send them back to the dry lab and they'll improve their model, and they'll produce a second batch of sequences. As well, we'll also begin presenting our work in order for everyone to gain presentation skills. In August, we'll do our last batch of iteration of new sequences. We'll obviously assay those as well, and then hopefully integrate them into a microbe, and then a batch bioreactor to actually see how our microbe would function in a sort of prototype of what we would hope to scale up to eventually. And then in September, we will finalize our work, prepare our speakers for their presentations in Paris, and also hopefully begin writing up a manuscript for preprint.
Yeah, great. So for dry lab, what we do for protein design is we use Python code to predict the function of protein sequences, and we use many interesting and really fascinating machine learning methods to explore this space. So a protein sequence fitness landscape space is very complicated and very epistatic. It's also discrete and it's huge. So machine learning is perfect for this task if you have sufficient data. So for example, here you're seeing the model predicting pretty accurately the thermal stability of some of the sequences.
So the wet lab's goal is to kind of validate the models and predictions that the dry lab comes up with, and then also to actually functionally integrate them into a microbe. So the left graph that you see is data from 2019. In blue, you can see, in light blue, you can see the wild type variant, and in orange, you can see the best variant at the time. And these are just relative catalytic rates. And then in gray, the last 5 variants, gray, yellow, dark blue, green, and purplish, I think that is. Those are all of our designs. And you can see that one of our rational designs called DIMI1 and one of the ML designs labeled TNU after the people who made them are actually quite close to the best designs of the time. So this assay that we will use to test our catalytic activity will be something that will be critical to our 3 iterative rounds of sequence testing and then going back to the dry lab to improve our model. And we're hoping that over these rounds of iteration, we'll be able to get really good sequences that are hopefully surpassing that in the literature. The other goal that we're planning to do with the wet lab this year is potentially a rational design campaign. So that will involve integrating a error-prone DNA polymerase into a strain of bacteria that grows solely on ethylene glycol, which is a byproduct of the degradation of plastic. So once we integrate our plastic catalyzing enzymes, we can perform a rational design because we will make mutations via the error-prone polymerase, and essentially over multiple rounds of selection, we will get strains that grow really well on ethylene glycol, and that will be because they catalyze the PET substrate from their media very well. So that's another way that we hope to evolve our enzyme to be quite good.
Yeah, so as Joseph said, we are striving this year for an iterative feedback loop between the dry lab and wet lab. So pictorially, that just means we're going to be generating some sequences using our machine learning methods. And then a subset of those sequences are predicted to perform well, are going to be synthesized in a lab. Here, I'm depicting it as an MD simulation, but it's like in real life, proteins move, and this is sort of representative of the wet lab, so to speak. And then the results of these experiments are going to be fed back into the model to improve the model and hopefully have it generate more sequences.
So the other team that obviously is extremely important to our work is what is traditionally called the Human Practices or Policy and Practices team. However, we're trying to rebrand that team as Business and Ethics. And so there will essentially be 2 goals of the team, and it will kind of be split in 2 sort of. We'll have one part of the team that's fully focused on business and entrepreneurship. And that team will be focused on designing business plans, looking for potential stakeholders, seeing how we can be competitive in the market. And this is an example from our business team last year who did some market research and made a basic business plan. We're hoping to build on that a bit. You can see that they talk about how we're innovative in the market, what makes us unique, what gives us a competitive advantage, the price of our end products, what we're selling, where we fit in the industry, and kind of the competition that we would be facing. So that's one part of the business and ethics team. The other part is the actual ethics and community engagement. So, of course, everyone thinks that breaking down plastics and finding better ways to recycle or reuse them is great, but to incorporate a microbial solution is a little difficult, and we have to find the right way to make the public feel okay about it, as well as make legislators encouraged by it. So this is the plastic science policy landscape for the government of Canada. So the ethics and community engagement team will really be involved with contacting legislators and community members and trying to engage them with our project, notify them of what we're doing, and give us feedback on how we can improve our project so as to better fit the needs of the people it will be affecting.
Right.
Oh, sorry, you can go about
Yeah,
this.
sorry. Yeah. So usually for these teams, their presentation is always very stunning. If you look at some of the examples from last year and the year before, in particular, this team, Ruperto Carolla, it's just incredible, the graphics and the effort that they have put in. In particular, I went to their presentation and you had all these amazing animations on their slides that really convey what their models are doing, what their project is all about. So this is really important for scientific communication because not only does it convey scientific ideas more clearly, really the idea is to make the general public know more about what we're doing by reducing a lot of the jargon and make it easily accessible. So we're going to have a big graphics team this year relative to last few years, and hopefully our output is also going to be effective in terms of communication. So this is a video that you can search up online, where this is our presentation from 2019. You can just watch it if you want. It's like 30 minutes. It basically describes all the work that we did in 2019 and that we hope to continue in 2021. And as Joseph mentioned, in May, those new members for Igem are going to participate in these CSBERG workshops. In particular, the dry lab and the wet lab will participate in SynB1, where they have curated these PhD students, graduate students, and OISE graduates have curated this curriculum for interdisciplinary learning, hands-on workshops for wet lab and dry lab, basically oriented around the Igem research idea. And SynB2 is for the business and ethics members, where they will talk about implementation sciences and things like qualitative methods and engaging with the community. Okay, that's the end of our slides. And yeah, if you have any questions, feel free to ask. Oh, hey, Gammon.
Hey, Tianyu.
Hello, Gamin.
Hi,
Hello.
Iris. Hi. Hi, Michelle. Hi, Abdur. Hope I pronounced your name right.
I'm going to stop the recording really quick.